Christina Monnier. The date is April 11th, 2024. So in this video, we're going to look at keratinocytes again. The lipid that we are going to talk about is a ceramide. The secretory protein is going to be the collagen, and the intracellular protein is going to be the keratin. So starting with the ceramide, ceramides are lipid catalyzed in the smooth endoplasmic reticulum and are metabolized further in the Golgi apparatus. How it arrives there depends on what kind of cell it is, for instance, mammal versus yeast, and I believe on what kind of ceramide, because there are thousands of different kinds of ceramides. In keratinocytes, a ceramide will travel for further metabolizing via COP2 transport vesicles to the trans-Golgi, or by the ceramide transfer protein to the cis-Golgi. While not all ceramides processed in the Golgi are destined to leave the cell, some are processed into lamellar bodies, which is what allows them to transverse the cell membrane during terminal differentiation. After secretion, these secreted bodies become key constituents of the lamellar structure, which is the barrier that provides structure to the stratum corneum, or the dead and dying outer layer of the epidermis. And now looking at collagen. So collagen is generated within the nucleus, and from here it moves to the surface of the rough endoplasmic reticulum, where its genetic code is translated by surface ribosomes. From here, it has been shown that the collagen then moves through a tunnel-like structure called the Golgi intermediate compartment. And that's how it gets to the Golgi apparatus for further processing. And in this process, the collagen proteins are packaged into transport vesicles. Collagen exits the cell through the process of exocytosis, whereby collagen's vesicles fuse with the plasma membrane and are secreted. This is an active process, meaning energy is required for collagen to transverse the plasma membrane. And after leaving the cell, it organizes itself within the lamellar body, or the lamellar structure. It becomes another key component of corneocyte's lamellar structure. So now looking at keratin, similar to collagen, keratin is originating in the nucleus and attaches to a ribosome on the rough ER, and then it's processed further in the Golgi apparatus. Instead of being packaged into transport or secretory vesicles, keratins are processed into microfilaments, which organize themselves around the cytoplasm, becoming a key part of the keratinocyte structure. Over the life cycle of a keratinocyte, these keratin filaments continue to populate the cell's inner structure until the cell dies and sheds from the epidermis. So by the time a keratinocyte becomes a corneocyte ready to slough from the epidermis, this anucleated cell is predominantly composed of this protein.